cual, ¿no?
Vale, vamos a ello. María. So, good morning. First of all, I would like to thank all the audience for attending the webinar. Today we have the webinar of the Regulation of Gene Expression and Genome Dynamics group. And this group is coordinated by Fernando, Monje and María Gómez. And they are going to introduce the speaker today. Thank you, María and Fernando. And if you want, you can introduce the speaker.
Okay. Well, hello, good morning, everyone, and thank you very much because this is my the first time I'm hosting one of these webinars. I'm supposedly going to replace Fernando in this group, which is not going to be an easy task because I'm sure he has done it wonderfully. So today we have a very young, talented scientist who is going to talk us about his recent work, Emilio Lecona. Emilio is currently working at the same institute where I am, at the Centro de Biología Molecular, Severo, Ochoa in Madrid. And he's a Ramón Cajal Fellow since 2018 here. And he started his career very close here also at the Universidad Complutense de Madrid with María Antonia Lizarde. Then he did a postdoctoral period at NYU and the lab of Danny Reinberg, a very fantastic expert on chromatin. And then in 2012, he came back to Spain with the Marie Curie fellowship to do another postdoc with Oscar Capetillo at the CNIO. And then he got un contrato Jóvenes Investigadores, which is a very prestigious project and grant, and worked there at the CNIO before getting as I said, the Ramón y Cajal Fellowship and coming back to joining us at the CMBO, when we are very happy to have him here. And yeah, well, you have seen the title of his talk is the role of BCP in DNA replication. And this is all I wanted to say now, and I give the stage to Emilio. Thank you very much, Emilio.
Thank you, Maria, and thank you, Fernando, for giving me the opportunity to present the work that we have been doing in the lab in the last years. And as Maria said, I'm going to try to tell you a couple of stories about how BCP, this protein, which is a wonderful molecular machine that is actually working in the recycling process in the cell, is actually regulating DNA replication, which is the process I am interested in. And sorry, now, so this, as you all know, you probably know, this is our textbook view of how DNA replication works, right? We have our machinery that is unwinding the DNA, with our helicase, and then we have DNA polymerases that are actually synthesizing the DNA in the leading and in the lagging strand. But this simplistic view was very much improved in 2012 when the group of David Cortez developed what it was called the IPRON, the immunoprecipitation of nascent DNA. And in this protocol, we could pull down nascent chromatin and compare it to mature chromatin, and then we can identify the proteins that are enriched in one and the other compartment. And with this method, in 2013, Andrés López Contreras in the lab of Oscar Fernández Capetillo actually discovered many new features of the replizome, which were not known at the time. And among them, I was very interested in the feature that I've been going to tell you about now. So it's the DEUBIQUITIN and SUMO landscape that happens to be in the replizome. What he found was that there is an enrichment in SUMO-related factors in the nascent chromatin, also modified by SUMO1 or SUMO2, and also an enrichment of ubiquitinated factors in mature chromatin. As you know, ubiquitin and SUMO are small proteins that are well known for targeting proteins for degradation, but they can also have many other functions. So with this in mind, we propose the following model in which there is a SUMO high and ubiquitin low environment in the replisome, and a SUMO low and ubiquitin high environment in the major chromatin. And I was interested in trying to understand how this actually works. What are the functions of this environment? How do we set it? And what are the consequences during DNA replication and for the replication stress response? In 2016, working in Oscar's Fernando lab, I described that there is a ubiquitinase, USP7, a ubiquitin-specific protease that works on SUMO and SUMO-related factors and removes ubiquitin, maintaining the ubiquitin levels low in the replisome. And this is important for DNA replication. Why? Because we found that when we inhibit or we delete USP7, what happens is that these proteins now become ubiquitinated and highly SUMO-related. And the consequence of this is a displacement of these proteins out of the replisome and a complete collapse of the DNA replication process. So inhibiting USP7 completely blocks the replication, initiation, elongation, everything, and then the cells cannot survive. So based on this work, then I wanted to understand better how this is working. How are these proteins actually removed from the replisome? What is the consequence of this ubiquitination and SUMOylation and the imbalance in this landscape? And with this in mind, I focus on VCP. And why VCP? So as I told you before, VCP is what is called as the recycling machinery in the cell. VCP is a homohexameric molecular machine that composes a ring and has a central port through which it unwinds and unfolds proteins through its ATPase activity. So basically what VCP does is recognize proteins in different cellular compartments to reduce, reuse, or recycle these factors by unfolding them and removing them from compartments. But how does it recognize the specific substrates? So to do so, VCP associates with a number, a slew of factors that are going to actually target VCP to specific proteins and different compartments. And they do so by recognizing the ubiquitination and SUMOylation on these proteins. So each cofactor will recognize ubiquitin and SUMO or both in different substrates and in different compartments. And it was also known at the time that VCP is involved in different stages of DNA replication. And there have been studies focusing on specific substrates for VCP in different processes, such as initiation, CDT1, termination, basically MCM7, and later work has shown that actually VCP is important to remove the replication machinery after DNA replication termination, and also in repair. But what I wanted to understand is, what is the role of VCP in this equilibrium during DNA replication, in the SUMO and ubiquitin equilibrium during replication? I had described that USP7 inhibition will imbalance this equilibrium and that there is a displacement of these factors. So I hypothesized that VCP was actually involved in removing these proteins, and it's important to maintain the ubiquitin and SUMO equilibrium during DNA replication. So with this in mind, I'm going to tell you two different stories. One of them is published, and one of them is what we're actually currently working now, and it's almost, I think, ready to be sent for publication. So the first one I'm going to tell you about is how we identified FAV1 as a cofactor for VCP in coordinating DNA replication together with USP7. And as a second story, I'm going to tell you how actually VCP is not limited to this specific setting, but it also regulates unperturbed DNA replication, and how I have discovered that VCP is important to maintain low levels of replication stress response in unperturbed DNA replication. So for the first part, as I told you, we wanted to analyze and understand what happens with VCP when we inhibit USP7. So this was our first question. If we inhibit USP7 and we have this huge accumulation of ubiquitinated and SUMO-related replication factors, what's going on with VCP? So we looked at the localization of VCP on chromatin using immunofluorescence after pre-extraction. So we are looking just here at the chromatin fraction, and we can see how inhibiting VCP induces an increase and an accumulation of this protein on chromatin, which is already described. Now, when we inhibit USP7, we have a further increase of the amount of VCP and chromatin, and this is even higher when we combine both of them. So the next question was, okay, so what is VCP actually doing here? How can we know more? So we just turned to proteomics to understand what happens to the interactions of VCP when we inhibit USP7. So we did cross-link proteomics, and we actually performed 2 different conditions to understand better the functions of VCP. We pulled down VCP after VCP inhibition, and here on the x-axis, we can find all the factors that are enriched when we inhibit VCP. And then on the y-axis, we perform USP7 inhibition, which is what we are actually interested in, to know what interactions are enriched when we block USP7. The first thing that came to that actually drew our attention was that we found SUMO2 as the main interactor after USP7 inhibition. As I told you, SUMOylation is characteristic for the replication environment, for the replication forks. So this was pointing to our role for VCP in recognizing these DNA replication factors. We also found the proteasome, and this is just to be expected since VCP targets ubiquitin-related proteins for replisome, for the degradation by the proteasome. Then, looking at DNA replication factors, we actually identified several components of the CMG helicase, again reinforcing the idea that we are actually seeing a function of VCP on DNA replication. But what we wanted to understand here was what happens with the cofactors of VCP. What are the cofactors that are actually working during DNA replication? And what we found was that among the cofactors, FAV1 and FAV2 are the most enriched when we inhibit USP7. So then we went on and asked us 2 different questions. The first one, do USP7 and VCP actually cooperate during DNA replication? So we can see that VCP is actually relocated to chromatin when we inhibit USP7, and it's interacting with replicating factors and SUMO-related proteins, but is it actually doing something during DNA replication? And the second question is, from FAV1 and FAV2, which is the cofactor who is actually driving VCP in this process? So for the first question, we just made a very simple approach. We took the currently known VCP inhibitors, since VCP is currently being studied as a target in cancer. So there are several VCP-specific inhibitors that we can use to block the activity of this protein. And we monitor DNA replication by following the incorporation of FDU. This is a nucleotide analog, and here we are looking at a high-throughput microscopy in which each dot is representing one cell. We can see how both USP7 or VCP inhibition reduce the rate of FDU incorporation by themselves. But in addition, when we combine both of them in the brown bars, we can see that combining both of them has a stronger effect in inhibiting DNA replication. So if we block USP7, then we block DNA replication, but if we block at the same time VCP, this effect is much, much stronger, indicating that indeed USP7 and VCP are working on the same process through different mechanisms. Now, the second question was, which one is the cofactor that is driving VCP in this process? And just to make a long story short, I'm just going to give you the right result, which is we only found an effect when we knocked down FAV1. So we actually depleted the different cofactors of VCP, and again, I said for DNA replication. And we found that similar to what happens when we inhibit USP7 and VCP, now when we have depleted FAV1, the effect of the inhibition of DNA replication by USP7 inhibitors is stronger. So you can see here in black how EDU incorporation is reduced by USP7 inhibitors and how this effect is stronger in FAV1 depleted cells. So with all this, we already said that VCP is cooperating with USP7 during DNA replication, and that FAV1 is also important for this effect. And we also had shown that somehow VCP is interacting with SUMO-related factors. So we wonder whether this SUMO-ylation, the SUMO-ylation of the replisome, is important for the functions for VCP during this process. So again, we look at a very simple question first: what happens to SUMO-related factors when we inhibit USP7? First, we look at VCP, and as I told you before, VCP, when we inhibit USP7, is accumulated on chromatin and is forming these big patches in the nucleus. And now, if we look at SUMO-related factors, which do not co-localize with VCP in unperturbed conditions, if we inhibit USP7, now we see how they relocalize to the same areas where VCP is found. So this tells us that VCP and the SUMO-related proteins are going to the same place after we inhibit USP7. And actually, I'm not showing it to you now, but we also know that replication factors also go to these areas. The second part was FAV1. So is FAV1 related to the SUMO-related factors? Because if FAV1 is actually driving the effects of VCP, then maybe FAV1 is recognizing SUMO-related proteins. And actually, there was a report 2 years ago showing that FAV1 could interact with SUMO-related factors modified by SUMO1. So we carried a very simple experiment. We pulled down FAV1 and looked whether we could recover replication factors and SUMO-related proteins. And as you can see here, in control conditions, FAV1 interacts with SUMO-related and ubiquitinated proteins, and also POLD2, which is one of the replication factors that is relocated upon USP7 inhibition. Now, when we inhibit USP7, we have a stronger interaction with SUMO-related proteins and also ubiquitinated proteins, indicating that FAV1 is actually driving VCP and interacting with SUMO-related replication factors. So now coming back to what we know about VCP and what we have already learned, what we propose is that FAV1 is somehow working as a SUMO sensor for VCP in these conditions. So reports VCP, it actually works in a way in which it can combine, it's like a puzzle. So it combines several cofactors to recognize different proteins. And it has been described in this paper by Herman Sindelin that actually can associate with UFD1 and MP-log together with FAV1. And this is what we actually think is happening. In yeast, it had been described that UFD1 or VCP could actually directly interact with SUMO-related proteins, but this is not conserved in mammals. The SUMO-interacting motifs are not conserved in mammals. So we thought that probably FAV1 is the module in this complex that is responsible for the recognition of SUMO-related factors, and this is why it's working on the replication. So we did 2 different experiments to prove that this is the case. We depleted FAV1 again and checked what's happening to SUMO-related proteins and VCP after we inhibit USP7. So first, as you can see, when we deplete FAV1, then we can see that there is an accumulation of SUMO-related proteins on chromatin. This is again immunofluorescence after extraction and high throughput microscopy. So we can see how in control conditions, depleting FAV1 increases the amount of SUMO-related factors. But not only that, when we inhibit USP7, we can see an increase in the accumulation of SUMO-related proteins on chromatin and a further increase when we don't have FAV1. Okay, so FAV1 is interacting with SUMO-related proteins. If we don't have FAV1, we have more SUMO-related proteins. The second part of the question is what's happening with VCP. If FAV1 is mediating the recognition of these proteins, then VCP recruitment should be also impaired. And this is what we found. When we deplete FAV1, we can see how the accumulation of VCP on chromatin induced by USP7 inhibition is now impaired. So somehow FAV1 is responsible to for bringing VCP to these SUMO-related and ubiquitinated factors. And with all these, we have proposed the following model, which is we know that USP7, we have described that USP7 is actually working on SUMO-related factors, removing ubiquitin and maintaining them in a low ubiquitination level during DNA replication. And now we see that at the same time, VCP together with FAV1, driven by FAV1, is also recognizing the SUMO-related and ubiquitinated factors, and it's actually extracting them when USP7 is not ubiquitinated. So together, VCP, FAV1, and USP7 maintain the right equilibrium between ubiquitin and SUMO, so DNA replication can proceed. And actually, we know that FAV1 is able to bind to both ubiquitin and SUMO-related factors thanks to experiments that we performed in C. elegans in collaboration with Thorsten Hopp. We also did different experiments showing that FAV1 can bind both modifications at the same time. And actually, this was a very nice collaboration. And in the paper that we just published recently in Cell Reports, we could also show that actually all this functional correlation in mammals is also conserved in C. elegans. We did some genetic screens. Well, we more like Thorsten did, genetic screens in C. elegans, and also confirmed that VCP and FAV1 cooperate with USP7 during DNA replication. And as I told you, I started this work while I was still in the Genomic Instability Lab, in Oscar Fernandez Capetillo's lab in the CNIO, and it was started by these two students, Patricia Oviedo and Pablo Vallador, an undergraduate and master's student that I supervised at the time, and then I finished it here at the CBMSO, and we published it some months ago. Okay, so this was the first part. Then the second part was, now we understand how VCP cooperates with USP7, but this is a very specific setting, let's say. So what happens when we inhibit USP7? This is not going to happen every time in any DNA replication cycle. But we also saw that somehow inhibiting VCP by itself has an effect on DNA replication. I have already shown you that. So we wondered, is VCP having any effect on an unperturbed DNA replication, an unperturbed S phase, without any exogenous damage? And before I go into this, I'm just going to give you a brief introduction on how the replication stress response works and how the replication works during an unperturbed DNA replication phase. So as you know, as I have shown you before, the basic mechanism in DNA replication is we separate the DNA duplex by the CMG helicase, and then we have Pol epsilon synthesizing the DNA in the leading strand, and Pol delta synthesizing the DNA in the lagging strand. And then we have Pol alpha, DNA Pol alpha, which is actually gonna set these primers for the elongation, but the processive DNA polymerases. And it's important to coordinate the progression of the leading and the lagging strand. So what happens when we have some problems and the progression of the polymerases is impaired? So when this happens, then we have an accumulation of single-stranded DNA. And this single-stranded DNA needs to be sensed and protected. And this is what happens with the proteins, with the RPA proteins, which are gonna just cover and protect this single-stranded DNA, but are also gonna lead to the activation of the replication stress response. And this replication stress response can be activated in two ways. In the first one, we have the RPA that is recruiting the complex ATR trip. ATR is the main kinase in the replication stress response. And then this single-stranded, double-stranded DNA junction that will recruit TOP-BP1. And TOP-BP1 is going to activate ATR directly. A second mode that was recently described, it's independent of the presence of this single-stranded, double-stranded DNA junction, and depends basically on the accumulation of RPA, and is mediated by this ETAA protein. So again, a trip will bind RPA and bring ATR. ETAA will directly bind RPA and activate ATR. The consequence of activating RTR is the phosphorylation of CHEK1 and the activation of CHEK1. That is the effector of this pathway that is going to actually mediate the cellular effects. And what are these effects? So although initially we focus on what happens, and all the studies were focused on what happens when we have strong DNA damage, now we know that this pathway is just as important during a normal S phase. So in an unperturbed S phase, we need basal levels of ATR and CHEK1 to limit the activation of CDK throughout the S phase. And why is this important? Because we need to stop the premature firing of origins that happen later in S phase. So thanks to this basal activity of the ATR and CHEK1 proteins, we're going to reduce the number of origins that are fired, and we're going to allow the replication to proceed during late S phase. Now, in the presence of damage, of course, this is much increased. What we have is a strong activation of ATR and CHEK1 that then leads to a blockage, complete blockage of CDK activity, complete inhibition of origin firing, and the activation of the G2M checkpoint. So cells will stop in G2M so that they do not go into mitosis with unreplicated DNA. Okay, so with this setting in mind, we wanted to understand what is VCP actually doing in these conditions when we don't have any damage. Is VCP important for DNA replication under these conditions? To try to focus on S phase, since VCP has many functions outside chromatin in many other places, what we did was to just synchronize the cells and just study the period of time in which DNA is being replicated. So we took cancer cells, we synchronized them using a single time eating block, then we released them and we treated them with VCP inhibitors in the middle of S phase. So as I told you before, VCP inhibition has impairs DNA replication. So here we are looking at the degree incorporation again, but in this case we are looking at it through flow cytometry. So we can also separate the different phases of the cell cycle. So here we measure DNA content with API. We have U1, we have all or most of our cells in S phase. And when we inhibit VCP, we can see how we reduce the amount of VTU that is incorporated. Okay, so next we thought, well, let's see if this is related to an activation of the replication stress response. So we looked into replication stress response. First, we said, let's see if VCP is necessary for the replication stress response itself. So we use hydroxyurea, but as you can see, can induce a strong phosphorylation of CHEK1 and DNA damage measured by gamma-H2AX, which is the phosphorylation that we use as a surrogate for DNA damage. Now we can see how, in the presence of VCP inhibitor, replication stress response works just fine. We don't need VCP for the replication stress response. But when we performed this experiment, there was also something that caught our attention, and was this increase, that is a slight but consistent increase in the phosphorylation of CHEK1 when we inhibit VCP. So when we inhibit VCP, we don't have a strong DNA damage, we don't have all this fuel replication stress response activation, but we do see always an increase in the phosphorylation of CHEK1. So we wanted to understand how and why this is happening. So the next thing that we did was to try to understand what's the consequence of VCP inhibition at the molecular level. And for this, we collaborated with Juan Mendez in the CNIO, and we performed DNA fiber assays. In these assays, basically, we follow the same protocol as before. We synchronize our cells, we release them, and then we treat them with the VCP inhibitor. And then we use two different analogs, nucleotide analogs, to mark ongoing DNA replication. Then we extract the DNA, we stretch the fibers on a cover slip, and then we detect each of the analogs through immunofluorescence. And this is an image of what you can see. And with this, we're looking at every single fork, and we can measure how fast the forks goes, so we can measure the fork rate, and we can also measure things like new initiation events or termination events. So using this approach, we analyze what happens when we inhibit VCP. And this is the result. So we can see how VCP inhibition strongly reduces the number of new origins that are found. So we can clearly see that VCP is necessary for new origin finding. And at the same time, we saw a slight decrease in the fork rate. And this, for the DNA replication aficionados, this is somehow a paradox, because what happens with DNA replication? So when the cells are duplicating the DNA, cells basically try to complete DNA replication during S phase. And to do so, they always maintain a balance between fork rate and origin firing. Meaning, if we have reduced origin firing, the cell will compensate by increasing the fork rate. And the opposite happens. If we increase the fork rate, then the cell will fire less organs. And this is not what we see here. So it led us to think that maybe VCP was having two different effects, one on origin firing and another one on fork progression. In order to analyze if this was the case, we played around a little bit with this balance. So we did two different interventions. The first one, we impacted on origin firing. We inhibited CDC7, and here we are blocking the firing of new origins. And then we'll look at the dynamics. And as you can see here, CDC7, of course, blocks DNA replication, the new firing of new origins. And when we combine both, this firing is still just inhibited just the same. As I told you, when we inhibit origin firing, we see an increase in the fork rate. And now we can clearly see that this fork rate depends on the presence of the activity of VCP. So this small decrease that we can see here is now clearly be seen when we increase the fork rate of the DNA replication forks. Okay, but to make sure that this is not just dependent on CC7, we made a different intervention. So we played with the other side of the balance, and we added nucleosides. The nucleosides, what they're going to do, they're going to increase the fork, and they're going to make the forks go faster by themselves. And again, we see a very similar thing. Nucleosides increase the speed of the forks. They concomitantly decrease the origin firing, and the effect of origin firing is not further decreased with VCP inhibition. And again, this increase in speed now clearly shows that VCP has a role in the elongation of the phase of the DNA replication. So with this, we have shown that VCP has a dual role in controlling both the initiation and the elongation phases of DNA replication. So based on this, then we want to understand how this is happening. So we thought, what VCP might be doing in the fork to actually control these processes. So we came up with some ideas, a couple of three ideas. The first and most obvious one would be that VCP is maybe just removing the blocks that are ahead of the fork. So if we remove the block, then the fork can go faster. Then we also figured that it might be actually working on the replisome, on the replication machinery. And by two ways. Maybe it's recycling the factors which we have shown before. We know that when we have ubiquitinated consumative factors, VCP is removing them. Or maybe it's helping in the coordination of the leading and the lagging strand by reusing the factors. So in this process, we know that we have to recycle polymerase alpha, polymerase delta, so we can have the Okazaki fragments going at the same time as the leading strand is progressing. And this idea was very appealing because at the same time that we look at the dynamics of DNA replication, we decided to look at whether inhibiting VCP could affect the symmetry of the forks. How do we do this? So again, looking at the DNA fibers, but in this case, in a slightly different protocol, we do two different incubations with different lengths of the nucleotides. And we can measure the ratio of these two nucleotides. We can see that in control conditions, this is more or less a third because the incubation with one is 10 minutes and the incubation with the other one is 30 minutes. Now, when we inhibit VCP, we can see that there is a strong increase in this ratio, meaning there is an asymmetry in the forks, there is some fork stalling going, and this is usually representing problems for fork progression and could be related to this coordination between the leading and the lagging strand. We have done these fork asymmetry assays using different measurements, and they all say the same. Inhibiting VCP induces fork asymmetry. And this was very interesting to us because in 2020, last year, the group by Luis Toledo had also shown that inhibiting pol alpha induces a very similar effect. It induces an increase in phosphorylation of CHEK1, induces an uncoupling of the leading and the lagging strand without a huge increase in activation of the replication stress response. So then we came up with the following idea. Maybe VCP is working on polymerase alpha, trying to keep this leading and lagging coordination. Well, so this was a very easy to test hypothesis. The first thing that we did was, okay, if it's removing, if VCP is actually controlling the presence of pol alpha and chromatin, it should be interacting with it. So we carried out reciprocal immunoprecipitation experiments. We pulled down pre-imase 2 as a component of the DNA polymerase alpha primase complex, and we can see how it pulls down pol alpha and how we can also pull down VCP, in particular in reads when we inhibit VCP, and this is something that is known with VCP inhibitors. If you inhibit VCP, you will stabilize the interaction. And the reciprocal is also true. If we pull down VCP, we can recover pol alpha, pol A1, and also prim2. And again, a stronger interaction when we inhibit VCP. Okay, but this doesn't mean anything other than they interact. We need to know if this is functional. So in order to understand that, we look at the chromatin fraction during DNA replication when we inhibit VCP. So again, we synchronize our cells, we release them into S phase, and then we'll look at what happens to pol alpha when we inhibit VCP. So here we can see how pol alpha, pol A1, and prim2 accumulate on chromatin when we inhibit VCP. And as a control, you can see that VCP is also accumulated. I have shown you before this with the immunofluorescence. Okay, so this means that VCP interacts and controls the presence of the DNA polymerase alpha primase complex on chromatin. At the same time, we know that VCP inhibition is also inducing CHEK1 activation. And then I wonder, well, are these two processes related or unrelated? Going back to the literature, it was very interesting for us to see that in 2000, the group of John Newport, using the Xenopus extract system, had already shown that the primase activity of the DNA polymerase alpha primex complex is necessary to activate CHEK1. So then we have now two pieces of the puzzle. We have BCP that is controlling pol alpha. The question is, since the primase activity is necessary for CHEK1 activation, is this related to the activation of CHEK1 that we see when we inhibit BCP? In order to assess this, we did two different approaches. The first thing that we did was to use pol alpha inhibitors. So like 2 years ago, 3 years ago, they found that some compounds were directly inhibiting pol alpha and they have anti-cancer activities too. So we made use of this directly, and we can see, as reproducing Luis Toledo's data, that we can see a slight increase in the phosphorylation of CHEK1. So what do we think is happening here? When we inhibit pol alpha, we actually block pol alpha and chromatin a little bit. We know that, we have done these experiments. And then we somehow increase CHEK1. So now what happens if we add VCP inhibitors on top of this? So what happens is that this basal activation induced by VCP inhibitors and the activation of CHEK1 by pol alpha inhibitors are much increased when we combine both of them at the same time. And we think that this is due to a strong accumulation of pol alpha and chromatin that is going to lead to the presence of these single-stranded, double-stranded junctions on chromatin. Okay, the second approach that we did was a little bit similar, but not the same. In this case, we depleted pol alpha. So you can see, using control, with a control siRNA, how the cells still accumulate phospho-CHEK1 when we inhibit VCP. What happens if we have less pol alpha and chromatin? Well, if we have less pol alpha in the cell, and now we inhibit VCP. So now what we can see is that the inhibition of VCP does not increase the phosphorylation of CHEK1 under these conditions. And we think that this is due to the fact that we don't have pol alpha, so there cannot be a further accumulation of pol alpha if we inhibit VCP under these conditions, indicating that the inhibition of VCP increases the phosphorylation of CHEK1 in a pol alpha dependent manner. So going back to our model, we see that VCP is controlling the progression of the DNA replication forks and somehow is controlling the presence of pol alpha on chromatin. And this is important to limit the activation of CHEK1. As I told you, CHEK1 is important to actually activate the checkpoint and induce the cell cycle arrest when the replication stress response is activated. So, as the last question that we wanted to ask is, if we inhibit VCP and then we accumulate pol alpha and then we activate CHEK1, do we have an effect on G2M? And this is very important because VCP inhibitors that were developed and are currently in clinical trials have been shown to induce an arrest in G2M. It was proposed that this is due to the effect on DNA replication termination. So people hypothesized, many groups hypothesized, that inhibiting VCP would lead to an accumulation of the replication machinery and impede the progression into mitosis. But now we have an idea that this might not be related to this at all, it may just be related to the replication stress response. So we came back to our basic scheme, but in this case, instead of releasing the cells for 2 hours, we released them for 3 hours, and then we treated them for 9 hours to allow them to progress into G2M and further into G1. We treated the cells with VCP inhibitors. We can see our slide where always consistent increase in phosphorylation of CHEK1. If we inhibit CHEK1 now, we see an increase in phosphorylation of CHEK1. This is always the case when DNA damage. And now, and somehow intermediate effect when we block both of them at the same time. Now, the important part is when we look at the cell cycle distribution, looking here at the DAPI, DNA content by DAPI, we can see DMSO cells, control cells, progressing to G1 after 12 hours. If we inhibit CHEK1 as expected, they have a lower, a shorter time in G2M, and they progress into G1 faster. As it has been described, VCP inhibition induces a strong arrest in G2M that can be actually overcome when we block VCP and CHEK1. So with all this, we propose the following model. So VCP would be actually working during normal, a normal S phase, an unperturbed S phase. And how would it be doing so? It would be controlling the presence of pol alpha and chromatin. And this would be limiting the activation of ATR and CHEK1. We also have data showing that this is mainly due to the pathway of TOP-BP1. We have done this experiment in collaboration with Luis Toledo. We have knocked down ETA and TOP-BP1, and we can actually show that knocking down TOP-BP1 reduces the effect of the inhibitors of VCP on ATR and CHEK1 activation. So this indicates that VCP, it's actually controlling the presence of Pol alpha on chromatin and the presence of these structures that activate TOP-BP1 and lead to ATR activation and CHEK1 activation. In this way, whenever the cell fires origins, then we're going to have an increase in replication stress that we know now that is necessary for the cell to actually control its phase progression. But we also have VCP counteracting this pathway and limiting the activation of CHEK1 to prevent the activation of the checkpoint. So with our data suggests that VCP is necessary for the normal progression of DNA synthesis during a normal S phase. And basically, as I told you, this is the group, the work of the group, chromatin, cancer, and ubiquitin system. The work that I have presented has been done by Rodrigo, a PhD candidate in the lab, and also by Alejandro, who did his TFG last year with us. Also work with the help of other people in the lab, Yolanda, Angel, Guillermo, and Álvaro, and of course the collaborators. So for this last part, Juan Mendez lab and Luis Toledo's lab, and for the first part, of course, the work we started in Oscar's lab in CNIO, and the wonderful collaboration with Thorsten. And I would like to also thank the CBM for all the help in the facilities and the funding agencies. And I will be very happy to take any questions.
So just remember to the audience that you can do your question. You can ask your questions to the speaker in the chat, both in the chat of the YouTube and also in the chat of the Zoom. I don't know, Fernando or Maria, if you have any questions to start. I have some curiosities, but if you start with this, because I am out of the field and probably this one, I'm very curious about the role of these proteins in cancer. You have commented something about the inhibitors, but if you want, I want to know a little bit more about this.
So VCP has been observed to be overexpressed in several types of cancer. Especially, it has been targeted in multiple myeloma. So basically, the idea behind inhibiting VCP was following the proteasome inhibitors, right? So proteasome inhibitors were developed, and they saw that they were active against multiple myeloma, but they also showed that at some point these inhibitors, they did not work. So people thought, okay, maybe we can inhibit VCP and induce prototoxic stress. And actually, there was an inhibitor that was in preclinical phases, but it turned out to have some effects on the phosphodiesterase. So now there's a second generation of inhibitors that is more specific, that is currently in clinical trials. Although the main focus has been prototoxic stress, in the last years there are several groups indicating that VCP might be important for replication. And actually, there's a very interesting factor. So disulfiram, the very well-known anti-alcohol abuse drug, is actually targeting one of the cofactors of VCP. And it has been shown that inhibiting this disulfiram, apart from working as an anti-alcohol abuse, it also prevents cancer development. And it has been proposed that is due to this effect on this, one of these adapters, actually one of the ones that work on chromatin and MPLOG4. So that's why it's in VCP is a good and interesting target, and why understanding the roles on the replication might also be important for the application of these inhibitors.
And especially for the side effects, no? Because you are explaining that probably it's it has a physiological role during replication, so probably there's
yeah, you have to consider that it could be impacting on DNA replication. And it's also important that to understand how it's inducing the block in G2M, because traditional chemotherapy that always uses or mainly uses the induction of DNA damage also induces a G2M checkpoint. And it has been seen that this C2M checkpoint also gives the cells the chance to become resistant, because it hits the cells to actually repair the damage. So this is also something to take into account when we apply these compounds.
We have some questions here from the YouTube chat, so I think that we can start by asking those to Emilio. Roman González Prieto, he's asking whether you have any idea of which are the proteins that get highly SUMOylated at the fork, or the E3 that perform the SUMOylation.
So the short question is no and no. And I would like to know, and we are planning some experiments to actually try to identify them. We may have some idea from the proteomics analysis performed by Ivo Hendricks. So we know that some components are highly SUMOylated in the presence of replication stress, but we would like to know what are the proteins that are SUMOylated even in the absence of replication stress. And we have some ongoing collaboration trying to figure that out.
Okay, so we have also here, well, Maria Moriel from Montpellier in France. She's first congratulating you, and then she has a lot of questions.
At least.
So hopefully, yeah. So she says that BCP is been known for its role as an extractase that removes proteins from membranes, thus shaping the protein of such membranes. It is now recognized that BCP is very important to shape the protein of the inner nuclear membrane. Then she follows up with both replication origin finding and fork progression have been linked in the past to the inner nuclear membrane, whereby the DNA anchor to it will condition the physiology of replication. And now the specific questions are, first, have you ever considered to explore the spatial subnuclear localization of the events you are presenting?
So we have not done a proper, like, let's say, immunofluorescence analysis for BCP and the factors. We see that BCP is actually not found particularly enriched in DNA replication foci in a normal S phase. So it's difficult to actually just look at the localization of BCP there, because we have BCP in other places. But I think it's a very interesting point that, to know what's happening with these proteins, and whether these, actually when we inhibit USP7, we see all these very specific pattern of reorganization of these proteins, and it might be related to the association with the membrane. So I think this is a very interesting.
Because she also asked that, I mean, and it's in this sense that I think it's a great question, is, do you think the phenotypes you show relate to the fact that inhibiting BCP may perturb the detachment equilibrium of forks to the nuclear periphery?
We do not see, I think I have looked at the foci, the replication foci after BCP inhibitors, and we don't see major changes. So I don't think we are affecting, but we should do some more experiments to actually confirm that.
So I actually
also have a question, but no, go on, Karen.
No, it's okay. But well, I have a couple of questions of my own. So first of all, is the first part you mentioned that you think that FAV1 is the main cofactor that is regulating BCP in this. And so I was wondering, in terms of the control of the activity of these proteins, whether, I assume that VPC is not, but whether FAV1 is cell cycle regulated. I mean, it's only expressed at certain, I assume that in S phase, but then that could be an explanation of why this cofactor and no others, if it's, you know, to give cell cycle specificity?
So actually, as far as I know, there is no cell cycle regulation of FAV1, at least in the expression level. I don't think people have been looking at the modifications. Maybe some modifications are changing. FAV1 works with BCP, but also has a role in pro-apoptotic pathways, and also works with BCP outside the nucleus. For endoplasmic reticulum stress and destruction of proteins from the ER. So my guess, it's probably going to be more than the expression. It's going to be probably the interaction at the level of the interaction and modifications that is driving FAV1 to work in one place or the other.
And just a curiosity, because you talk a lot about inhibition, but what happens if these proteins are overexpressed? I mean, especially I assume FAB1 that is bringing VPC to the chromatin.
So actually, I haven't tried that. I haven't tried the overexpression of FAB1. Actually, I have done some experiments, and cells don't like to have too much FAB1, but this might be related to the pro-apoptotic activity.
Okay.
So it's difficult to separate that there.
Maria, I think you.
Yeah, I also have a couple of questions, Emilio. Well, first of all, thank you for the very interesting talk. So can you rule out that VCP FAB1 effect is only occurring in other chromatin proteins that are not necessarily at the replication proteins? I mean, maybe histone recycling, things like that?
No, we have not actually ruled out that possibility. We know, as you can see in the immunoprecipitation studies that I showed, that actually FAB1 pulls down replication factors, but also histones. So it could be well both. I don't think it's necessarily one or the other. I don't think VCP has been involved in removing histones from chromatin, so it would be more histone chaperones. And I don't think that VCP has been associated to histone chaperone, but I'm not sure about it. I think both things can be anyway important, because there's a paper that was published 2 days ago showing that SUMO of linker histone H1 might be important for chromatin structure. So that could be also a possible substrate, more than core histones, more related to histone H1. But we are trying to.
We should talk about that then.
We're trying to identify FAB1 substrates. That's our next move. We're trying to identify FAB1 SUMO-related substrates.
Yeah, that was another question. And the other one is relating both stories that you told us today. So is the VCP interaction with Pol A related to the SUMO or UB modifications of Pol A, or is it a direct interaction? What do you think?
The 4 subunits of the DNA polymerase alpha and the primase complex are actually have been described to be either ubiquitinated or SUMO-related in proteomic studies. And this is basically our next step, to try to identify the ubiquitination and SUMOylation events on this complex that could be driving VCP there, that could be actually recognized by VCP.
So is it known if they are dynamically changed through progression, or in, I don't know, in the leading versus the lagging strand or anything like that?
So it's known that they are modified, but there's nothing known other than the sites of the modifications.
Okay.
Okay.
Don't see more questions.
No, in the chat.
So,
Not in the chat.
not in the chat. So if you don't have more questions, I would like to thank again the audience for being with us during these webinars. And also I want to thank Emilio for the fantastic talk, and also Maria and Fernando for organizing the webinar, and in particular Fernando, that is leading us, and he has done fantastic work during this year as a coordinator of this group. And I hope to see all of you in the next webinars. So thank you, all of you.
Thank you.
Thank you very much.
Thanks.